Phoebe Jekielek is going to be our next presenter. She was an educator and researcher at Hurricane Island, just finished a PhD at University of Maine, and is currently a postdoc working with Damien Brady. And I think her talk is going to kind of be a blending of the two. She's going to present to us. Unfortunately, Phoebe's not here, so she's challenging my major kryptonite, which is getting technology to work. So luckily, there are people up here who are gonna do that, and Phoebe's going to join us via Zoom.
That's looking pretty meta. There we go. Oh, hi. And I did share my screen, but I can see that you can only see my big face. Do you want me to try and reshare?
We'll get there.
Okay. Hi, everybody. Yep, I'm having some serious FOMO up here in Maine, missing this amazing opportunity. But I'm so glad that I'm still able to kind of share some of what we've been doing up here with all of you. And yeah, I'm sure it will lead to some really good discussions. So my name is Phoebe Jekielek. Today, I'm going to talk a little bit about some life history monitoring efforts that we've been doing up here in Maine as part of a few collaborative projects with both scallop fishermen and aquaculturists. And I have a lot to get through, so I'm going to try and keep myself on time. So briefly, in Maine, our inshore state managed fishery brought in over $9.3 million in 2023. It supports around 305 draggers and 31 divers, and it's managed in 3 zones along the coast. Zone 1 essentially goes from the New Hampshire border to the middle of Western Penn Bay, where those purple and green lines meet. Zone 2 spans from Western Penn Bay all the way down to Cobscook. And then Cobscook Bay is its own kind of magical management space way down there in eastern Maine. And alongside this established, economically lucrative and healthy fishery, we have a growing aquaculture industry. This map shows the locations of all those existing aquaculture sites that are spread along the coast, or you could think of it as that are spread within the wild harvest management zones for scallops. So of these leases and LPA sites, around 14% of them list scallops as a species to grow. At this time, and to my knowledge, as you heard from Sydney, there's less than 10. I know there are around 5 farms that are growing at any scale to actually sell, which are represented by these red stars. Please speak up if this is incorrect. It continues to be kind of a moving target around who is actually growing scallops and selling them. There are some other farms growing scallops, but we don't actually know that number, and most of them are growing them in very small amounts. So the point is that there's a lot of potential for the growth of scallop aquaculture in Maine, but it's not yet fully realized. And what we end up with is this overlap of wild and farm scallop populations in certain areas, especially in Penobscot Bay, with the potential for this overlap to expand along the entire coast of Maine if more people start growing scallops at any scale. And so it also provides a really unique opportunity to compare these populations, wild and culture populations, test new methods for monitoring them, and deepen our understanding of how they might interact. So for example, as we've heard, in Maine, the emerging scallop aquaculture industry relies on seed collection from wild populations, as there's not a scaled hatchery success yet, which is a key point of interaction. And some other specific opportunities for potential beneficial interactions of wild and cultured shellfish populations include the potential for larval spillover, or what I'm kind of calling passive enhancement, from farms or closed areas to support wild populations. There's evidence of larval spillover from mussel farms supporting wild populations in New Zealand, and we can kind of perhaps assume that this is happening from our farms in Maine, but we're not yet fully understanding this potential. And there are some new research efforts looking into this in the next couple of years. There's also a big potential for post-larval capture, culture, and release to enhance wild populations by what I'm kind of calling active enhancement opportunities. And we're going to hear more about this later on today. And so again, a common need toward evaluating these potentials starts with a clear understanding of larval and reproductive processes. I did want to put this up there. I just added this because I know that Nate mentioned the circulation in the Gulf of Maine. The EMCC is the Eastern Maine Coastal Current. It hits Penobscot Bay. From the outlet of the Penobscot River, it kind of gets shot out here into Jordan Basin and offshore, but then also part of it continues into the Western Maine Coastal Current That's all I'm going to say about that now, but that is something to kind of keep in the back of your minds in these conversations, and especially with some of the data that we're going to see coming up. And then also just Mid-Coast Maine is here where I am. I'm actually right over here, almost at the little point of that P. And y'all are down here in Rhode Island. So we've seen this a couple times, see scallop life history. So in order to kind of really understand how these populations can interact, we need to monitor reproductive and larval dynamics. And neither of them are being monitored on a large scale in Maine so far. So today I'll be talking about results from 2 studies that are attempting to do that, or at least start to do that. First, I'll talk about a coast-wide collaborative effort to monitor temporal and spatial larval dynamics in Maine. And then the second will highlight some results from an effort to monitor spawning and reproduction in wild and cultured populations. So diving in, this is a collaborative project between fishermen, farmers, and four organizations: the Maine Center for Coastal Fisheries, Colby College, Hurricane Island Center for Science and Leadership, and the University of Maine, to really understand larval distribution. I want to first off give a major shout out to the industry collaborators. The work for the rest of this presentation for both of these projects is not possible without industry collaborators. I actually don't know who's there, but if you see any of these people or hear any of their names, please corner them, as it has been said that people will be cornered down there. They can speak to the work that we've been doing. And also I know that Carla Genther is there today in the crowd, and Anya Hoppel will be there tomorrow. So these are other people that are present that you can talk to about these projects. But again, not possible without fishermen, farmers, and also land-based organizations and businesses that allowed us to do the processing for this spat work at their sites. So the objectives of this project are to overall increase our understanding of larval distribution in Maine through evaluating the spatial and temporal variability in larval supply, attempting to develop a low-cost replicable monitoring protocol to engage industry, strengthening relationships and knowledge sharing between fishermen and farmers, and also providing authentic education for high school and undergraduate students. The way that this all started, in summer 2022, we identified 4 transects along the coast in a series of in-person meetings with fishermen and farmers, and also informed by a science advisory committee. Those transects are, oops, I see I have a bunch of bags on Alex and Mike Murphy there. Sorry about that, guys. But those transects are Casco, Muscungus, Jericho, and Narraguagus Bays, spanning the entire coast of Maine. In 2023, the transects consisted of 5 sites within each of these transects, with the exception of Muscungus, which only had 3. And these sites are trying to be set up inshore to offshore, with paired lines of spat bags here deployed at each site. And each line is equipped with 10 bags. And you can see I added a couple extra bags to this figure from Dana's from a Scallop Aquaculture publication. And you can see the total lines for each transect in 2023 along the coast. And then in 2024, and again, I'm sorry, I should say that all of these transects for the first year of deployment were within the 3-mile state line. And in 2023, 2024, we got additional funding, and we added 2 sites in federal waters to each of these transects. And so that expanded our line numbers along the entire coast. Each line was also equipped with a hobo temp logger, and 1 inshore and 1 offshore line in each transect was equipped with a salinity logger. Just to give you a kind of a quick look at how we deployed these. So in the first year, in year 1, in 2022 and 2023, we had 2 deployments. So we deployed in October, a whole set along the entire coast. We retrieved all of those and processed all of those in January. And also at the same time, we put out a whole other set of lines and bags at each of these sites in each of these transects, which were then collected and processed in the spring of 2023. In years 2 and 3, so last year and this year, we have a single deployment we put out in the fall and we retrieve in the spring. When we collect the bags, in order to make processing a little simpler, like I said, we had 10 lines on each, or 10 bags on each line. We combined the top 5 bags and the bottom 5 bags to really try and help the process go faster. There's a lot that comes back on these bags, as we heard from Nate. And it can take a long time just to get through 5 bags. So we essentially had garbage cans full of water where we emptied the bags and the Netron into, and then we meticulously picked any additional small scallops off of the bags and the Netron and put them into those samples. Then we sifted and sorted these samples through a series of sifters. And then we weighed them to get, and we also picked out the biofouling that was on anything that was in there. We did our best to pick out as much algae and other organisms that were in there to get as clean a sample as possible. Then we got a total weight of that sample. And from that sample, we took 3 1-gram subsamples in order to conduct counts so that we could then estimate a number of scallops from each of those samples of 5 bags. We also took samples for hopeful future genetics work and to understand length frequency distributions. So what did we find? So first off, gear loss is an issue. This is definitely something that we have to consider in the future. Like Nate said, there are some places where you should put gear and some places where you shouldn't. Last year, we had those big storms, which were devastating up and down the coast. So that is something I want to share from the very beginning and to consider when looking at this data and thinking about these efforts in the future. Gear loss is an issue. There's high interannual variability. So what we're looking at here is the total estimated spat count. And this is per transect. So along the bottom, you have the transect. Blue is 2023, red is 2024. And then the numbers above each bar represent the number of bags counted from that transect. And so we see both high spatial variability in spat counts and high interannual variability in these counts. And the Blue Hill Jericho Bay transect has its highest numbers in both years. And it's worth noting that the 2023 totals all came from our first retrieval in January. That second deployment that I mentioned that we put out between January and April/May, there were about 25 scallops total in that entire haulback. So there was very little settlement between January and April, another point of discussion for the future. When we take those totals and break them down into lines within each transect, we again see high spatial and temporal variability. Here are the transect areas from west to east and the lines across the bottom, A1 through G2 from inshore to offshore. So just a reminder that we increased the number of sites in our 2023-24 deployment and added those federal sites. So sites F1 through G2 were not sampled in our initial deployment. And again, what we find is high spatial variability, both along the coast, so from west to east, and also within those transects from inshore to offshore. And again, that interannual variability. And in this figure, what we're looking at, each dot represents the total estimated spat from each of those 2 5-bag counts from the line, from each line within a transect that we recovered. And this data is only from 2023. And so remember that we separated each line of 10 bags into sets of 5 to make processing easier. And along the x-axis, we have the depth at which the line was deployed. I know someone asked a question about depth earlier. So what we found is that this line depth explains around 58% of the variation in spat count from these bags in 2023. Again, we haven't looked at our most recent data from this past year, but we will have another third year also to do this work with. So we do need to explore this relationship further. As I mentioned, we also have temperature and salinity data from these deployments. This is a very preliminary data, but what I see are more obvious temperature differences from inshore to offshore within the Western transects. I realize that these are kind of small at the top, but this is Western at the top and Eastern on the bottom. So we see more obvious temperature differences from inshore to offshore in the Western transects in comparison to the Eastern. And there's a lot more to look at with this temperature and salinity data, which I'm not showing any salinity data today, but there's much more to do to understand the spatial and temporal differences and how the environmental variability interacts with those. And so we would love to talk with people if you work with temperature and salinity data and have some ideas of how to visualize, how to analyze that. We would love to chat with you. And I also want to mention this because I believe it speaks to the opportunities projects like this offer for engagement with the broader fishing, farming, and local communities. They're key opportunities for education. And it also speaks to the massive numbers of hands that this work takes to make successful. I haven't done the actual count of how many hands have touched a baby scallop in this project in the past couple of years, but it is very, very large, and it is a very large lift to do the processing, deployment, and analysis of all of these bags every year. So worth a mention. So takeaways so far: gear loss is an issue. This could be because people analyze gear, or I'm sorry, they deploy gear differently. As Nate mentioned, people do it differently in different areas. There are different abilities of people to check their bags over the years. Some people are deploying in areas where they don't actually fish through the winter. So that's something to consider. There's interannual and spatial variability, and it's high. Sometimes orders of magnitude difference between sites and also between years. There's an interesting relationship between depth and spat. So we need to understand the role that distance from shore might play in spat distribution a little deeper. And there's also high spatial environmental variability. And we don't yet understand the influence on the interannual and spatial variability that this environmental variability has. So that's all future work to be done. I'm gonna skip this and just kind of keep moving because I know that I'm tight on time. So I'm gonna move into some patterns that we've seen in reproduction in wild and cultured populations. And there's a real opportunity to better understand some of these patterns in larval supply if we can connect these two things. Again, really huge shout out to Marsden and Bobby. I'm not sure if Bobby is there, but it sounds like that Marsden is there. And also Hannah Twombly and Connor O'Neill. I don't know if they're there, but the following work would not have been possible without all of these people here choosing and allowing us to access their farms and collaborate with us for 3 years of research. So setting the stage for why we might see potential differences in wild and cultured populations and why it's worth asking that question. Scallops grown in aquaculture are grown in lantern nets. These lantern nets hang from a horizontal long line suspended in the water column. Some people are doing them by ear hanging. I know that Andrew Peters is there. You can ask him about that, but same idea. There's a horizontal long line that's suspended anywhere from, 2 to maybe 5 fathom below the surface of the water. These nets generally have high densities of scallops in them, and because they're up in the water column, they're not accessible to predators. Whereas comparison to our wild scallops, they live on the bottom in sandy or shell-hashed habitats, they're found in lower densities, and they're fully accessible to predators. And so these two environments can have very different flow, food, and light regimes because of these different parts of the water column that they're inhabiting, which sets us up for a question asking, what does reproduction look like in wild and farm scallops? So for this work, we partnered with we did the research on 3 scallop aquaculture sites in Penobscot Bay. Up here in North Haven, Connor and Hannah's farm. Over here in Stonington, Marsden and Bobby's farm, and then the Hurricane Island farm down here off of Hurricane Island. This was a multi-year study with weekly sampling frequency between June to October from 2020 through 2022. And at each of these sites, we collected shell height, wet weights of total viscera, adductor, gonad, and shell weights from farm populations, as well as we collected using scuba from wild populations near to each of these sites. We also collected some environmental data at each sampling event. This figure shows a sampling frequency of farm to wild populations, and just orienting you to the fact that the data you'll be seeing in the following slides focuses on the size classes with the highest sample sizes from both wild and farm populations. And those are denoted with an asterisk. So essentially 80 millimeters to around 109 millimeters. This is also relevant to the wild harvest fishery because the legal harvest size is around 101. And so you can think about this as also some seed scallops feeding into the wild harvest fishery and harvestable sizes. Oop, there we go. So on the left, we have our parts. So from our dissections, the adductor, gonad, and viscera. And what we're looking at is the morphometric comparisons of wild, which are represented by the light boxes, and farm scallops represented by the dark boxes of the meat, gonad, and viscera for these 3 different size classes. The asterisk denotes a pair that is not significantly different, which means that the meats, gonads, and viscera of farm scallops were larger than wild scallops. I'm sorry, were heavier than wild scallops during the spawning season for all 3 size classes, except for the gonads in the smallest size class. In contrast, wild scallops had shells with greater masses than farm scallops for all size classes. We can also see this when we look at the relationship between the scallop component mass, the mass of the meat, gonad or viscera, and the total mass of the scallop. And so we see these relationships again. Green are farmed, yellow are wild. Again, the asterisk shows not significant. So again, we see that the overall mass for meat, gonad, and viscera were greater for farm scallops than wild scallops, with the exception of the meat weight for 2021 and 2022. And the shell mass to overall mass was less for farm scallops than wild scallops. So what this means is that farm scallops have meatier meats, bigger gonads, which potentially translates into greater reproductive output, and that they overall allocate more energy to body tissue in comparison to the wild counterparts. When it comes to shell, wild scallops have heftier shells, possibly because they need to allocate more energy to defending themselves in the wild. When we look at spawning, we use the gonatosomatic index. So the GSI is on the y-axis, and on the x-axis, you have dates of sampling. Shows the average GSI at each sampling event at the Hurricane Island and the North Haven farms. And what we see is that wild and farm scallops generally spawn at similar times. And peak spawning is indicated by the peak mean GSI, followed by a continuous decline of mean GSI. Didn't really differ more than 3 weeks within years and occurred in late August and early September in each year. What we also see is that peak spawning coincides with peak temperature, which is represented by these dashed vertical lines. So spawn timing does not seem to be greatly impacted by farming scallops. And farm spawning seems to mimic the natural populations, which could be good for this idea of passive larval enhancement from farms. Lastly, farming doesn't seem to affect spawn timing, but it may affect synchrony. So you see the gonad weight condition in yellow on the left y-axis, and meat weight condition in blue represented on the right y-axis, and then the black line represents temperature. And what I want you to notice is that farm populations have a taller, narrower peak in comparison to their wild counterparts, indicating that there's potentially more synchrony in spawning populations of farm scallops, perhaps due to these high densities found on farms. And so kind of this message to get busy reproducing gets delivered more quickly because of these high densities, and perhaps because of the environment that farm scallops are experiencing. So in short, energy investment is altered by farming. And what that means is that aquaculture provides a bigger product for meat selling and potentially offers an opportunity for enhanced gamete production. When it comes to life events, spawn timing is not really affected, but perhaps synchrony is affected. And what this might mean is that farming can enhance reproductive investment and potential larval output. Lots more work to be done here, but perhaps this might facilitate farm and wild genetic connectivity. There are some questions to be asked around what that larval supply from farms could actually look like and what it could mean for the future of passive enhancement from farms. And what does it mean for the genetic diversity of farmed and wild populations? So, few things, last comments. Industry engagement has been essential for these 2 projects. And I imagine that it's going to be essential moving forward. They have been huge knowledge sharers, informing basically every part of the process for both of these projects, and it would not have been possible without them. So we need industry knowledge, input, and expertise and experience to make these successful. An ideal thing to happen would be to model and connect some genetic work to tie reproduction and larval supply together, and really understand what these connections to the adult populations look like. It would be wonderful to expand these monitoring efforts to see if these patterns apply more broadly and identify additional questions that we can ask. Again, they take a lot of hands, they take a lot of work. And so expanding these efforts is difficult, but I think worth discussing. We need a better understanding of the environmental and oceanographic dynamics on both reproduction and larval distribution. And more questions to be asked around passive versus active enhancement considerations based on these findings. So can increased reproductive output from farms be meaningful to wild populations? And when we think about seeding areas, so potentially growing scallops in aquaculture and then seeding them, what does it mean if scallops grown in aquaculture have lighter or potentially thinner shells? Does this make them more susceptible to predators? This is just, I was going to try and come back to the guiding questions, but I see how far past I am, so I'm going to move past those. But I think this work ties into these guiding questions pretty well. And again, just a massive thank you to everyone that has been part of these projects, and also to all of these funding sources that have made these projects possible. So thank you very much.